and welcome to Teach Me in 10. In this episode, we're joined by Dr. Cody Palumbo, Senior Application Scientist in Technical Design at TriLink BioTechnologies. Cody will discuss why high-purity guide RNAs, also called gRNAs, are essential for achieving precise and reliable CRISPR genome editing. He'll explain the differences between desalt and HPLC purification, when HPLC should be used, and how TriLink is helping researchers in the biopharma space access consistent, high-quality gRNAs to accelerate their work. Let's get into the video. Hi, Cody. Thanks so much for joining us today. I'm looking forward to hearing how to maximize CRISPR success in biopharmaceutical research. So to start, can you outline how CRISPR is used in biopharmaceutical development?
Yeah, absolutely. So CRISPR has been really transformative for the biopharma industry in general. Um, so there's kind of three big buckets that it's really made a big impact. So the first one is in therapeutics, the first one. And so this is where you're trying to use CRISPR as the actual therapy. You can think of this as changing genetic mutations. Um, you see this in ex vivo cell therapy, where you're generating engineered CAR T cells, um, as well as the field as a whole is sort of moving towards in vivo gene editing as well. So this is directly delivering the CRISPR machinery to cells to do its work. So the second big bucket then is engineering cells, where you're trying to maximize production or you're trying to create a disease model. For example, creating a disease mutated mouse model so then you can go study it. You can now enable that much quicker with CRISPR. And then finally, genetic screens. So there are um, you can go do a huge CRISPR screen, find a new protein of interest, a new gene of interest, and now you have a new target. And then downstream, you can create, a small molecule or biologic to hit that. And so those are really the three large buckets that folks are using CRISPR for now.
Amazing, thank you. And then I was wondering if you could talk a bit about guide RNAs or gRNAs and, for example, how are they produced?
Yeah, absolutely. So yeah, guide RNAs are the honing device for CRISPR. So that is what is directing the machinery to the loci in the DNA that it will eventually cut or direct the machinery to do some other cool things. Um, yeah, largely now these are produced by chemical synthesis. Um, and so essentially we start with one nucleotide on a resin, and then we add like Lego pieces, one nucleotide at a time until we grow the guide RNA all the way out to, in general, 100 nucleotides or longer. The big advantage here is that you can add chemical modifications. And we have a whole variety of those that we can add here at TriLink. And as the field is moving more towards in vivo gene editing, those chemical modifications are becoming more and more important. And so chemical synthesis gives you that great advantage of doing it.
Amazing, thank you. And the next question I have for you is, what's the difference between desalt and HPLC purification?
Yeah. So in general, there's always been these two buckets of purification. Desalt is the most basic. It's the most cost-effective way. Essentially, it is a purification technique after synthesis and deprotection that is really only meant to remove salts that come off and small molecules. So it's a very quick cost-effective way of kind of cleaning up guide RNAs, so they wouldn't interfere necessarily with your downstream application. HPLC is really a polishing step. So HPLC purification, you would be removing truncations, synthesis impurities, really enriching for the full length guide RNA that you want with all of the chemical modifications that you want. Um, and so really it's always going to be a balance between, fast and a little bit more affordable and highly pure very, very enriched for the desired product.
Right, I see. And could you talk a bit about when someone should use HPLC purification?
hmm Yeah, we get this question all the time. So I'm glad you brought it up. So in general, as you progress from screening to lead candidate selection and then finally onto the clinic, there's this a break point, and everyone's break point's a little bit different. But in general, as you are finalizing your lead selection for your candidate for your guide RNA, that's really when you want to make that change over. So that's when your consistency is going to go way up, from lot to lot, or just you can, rest assured that like this is now pure. It's removed a lot of those impurities. Whereas desalt is really meant for screening to try and, weed through all of the different guides to find that special one for you. And so that's in general the advice of when you want to switch over is I would say once you're finalizing lead, and then definitely for moving into talks and things like that, and of course GMP, and those things will be HPLC purified as you go through.
Amazing, thank you. And then lastly, to wrap up, can you explain how TriLink is making high quality gRNAs accessible to researchers in the biopharma space?
Yeah. So there's really two main fronts that we're pushing. I'm happy to announce actually, we just launched a ligation based method for CRISPR screens. So I mentioned here before, you've always had to choose between speed with desalt and high quality with HPLC. We've sort of split the difference now, and we've created a ligation based method. So you sort of get the benefit of high purity and well to well consistency in your screens. Um, and it's much quicker than trying to get HPLC purified things. So, but you also get the benefit of cost savings. So it's sort of how we're trying to push the needle a lot more forward in the CRISPR screens, so that you're not getting false positives, false negatives, unintended off-target editing when you're doing those screens. And then I would say the next big front that we're pushing on is highly modified guide RNA. So I mentioned this, we're moving towards, the field is moving towards in vivo gene editing, really, where stability of the guide RNA is becoming more and more important. And so we do offer, um, as many chemical modifications you'd like to put onto the guide, we can definitely support that. And we don't shy away from that. And then on the back end, when you are getting ready for HPLC purification of those, we've developed specific methods to ensure that the purity remains high as well.
Amazing. They sound like really important developments.
They are.
Thanks so much for providing such an interesting insight into maximizing gRNA quality. It was great to talk to you.
Yeah. Thank you.
Thanks. Thank you for joining us for another episode of Teach Me in 10, which was brought to you by TriLink BioTechnologies. TriLink delivers pioneering nucleic acid tools and services to help bring next generation research to patients faster. To learn more about TriLink's gRNA synthesis services, explore resources, or request a quote, check out the links in the description. As always, if you enjoyed this episode, don't forget to like and subscribe to catch all our latest releases.